Welcome to In Plain English, a podcast where we discuss scientific research in terms that are accessible to everyone, not just the experts. I'm your host, Jamie Moffa. Before we get started, a few quick reminders. You can download the paper for each episode at inplainenglishpod.org by clicking on the episodes link in the main menu. We believe in open access science for all, so the papers we choose will always be free for you to download. If you have a question or comment about the previous article, you can submit it under the Continue the Conversation tab. If you are interested in being a guest or presenter for a future episode, you can click on the Become a Guest tab on the website. You can reach out to us on Facebook or Twitter at Plain English Sci and subscribe to our podcast on Spotify, Google Podcasts, or wherever you get your podcasts so you never miss an episode. This episode will be our last for 2021. A huge thanks to all our guests so far, as well as to all of you for listening and spreading the word to your friends. Because of the holidays, we won't have a regular episode in January, so our next full episode will be in February 2022. However, I am working on a compilation of fun conversations that got cut from the regular episodes, so be on the lookout for that sometime in early January. With that out of the way, on to today's paper. On today's episode, we will be talking about how hormones affect the growth and spread of prostate cancer. Presenting this topic is our expert guest, Ralph White. Ralph, would you like to introduce yourself?
Heck yeah, well, hi everyone. I am Ralph White III. I am a fifth-year PhD candidate in the Department of Pharmacology at the University of Minnesota. Pronouns are he, his, him. I also can be deemed a chicken nugget sometimes, as much as I eat them. But happy to be here. I will not say I'm an expert, but I'm a learning expert. I'm trying to get into it.
Awesome, thanks, Ralph. Joining Ralph for this discussion are our two guests for this episode, Maura Lydon and Alexa Ruel. Would you each like to briefly introduce yourselves?
Sure. I'm Maura, she, her pronouns. I am a librarian at the St. Louis Public Library, and I know just enough science to get me into trouble.
My name is Alexa Ruel. I'm a PhD student at Concordia University in Montreal, Canada. I study decision making, so this is far out of my area of expertise, but I'm really interested in talking about it today. Pronouns are also she and her.
Great. So without further ado, Ralph, take it away.
All right, y'all. So I'm going to give you the laydown of how I think we should go about it. We're going to hit main points today. And I love how Alexa already said, just like, this is way out of my field. It's kind of out of my field too, because my studying is involved with prostate cancer. So does anyone really know how prostate cancer works, or in terms of what signaling is happening in prostate cancer?
Only as much as I know how any cancer works, so it's like cells that are not doing their job. That's about it.
I'm on the same page.
I love it. I love that Maura and Alexa said the main thing. Cells had one job to do. Well, they have a couple of jobs to do, but they have a major job of just keeping stuff together. And this is just going to help you all in terms of why I selected this paper, is when it comes to prostate cancer, a lot of the time you focus in on a particular signaling pathway called the androgen receptor pathway. What happens is, and this is for everyone, every person has androgens, every person has estrogen. The question is, how much do you have? So for most men, they have primarily androgens and less estrogen. However, as they grow up and get old, estrogen starts on the rise. But we don't have to get into that anymore. During this time, androgen will be then converted into a more potent version of it. In terms of this case, it could be testosterone, but then it'll go from testosterone to the thing called dihydrotestosterone, or DHT. Just think of it as like this particular molecule had a growth spurt and was like, oh, I can do everything now. And so it'll bind to something called the androgen receptor. This will then go down a cascade of, let me link up with another androgen receptor that has a bound DHT molecule with it. And then it will bind to DNA in the nucleus, and then it'll go through AR target gene expression. That's what usually happens. However, for today, we're talking about a paper that doesn't even focus on androgen. It focuses on estrogen. The paper we're talking about today, you all, is called Estrogen Receptors Promote Migration, Invasion, and Colony Formation of the Androgen-Independent Prostate Cancer Cells, PC3, through beta-catenin pathway. A lot of words, but mainly what we're saying is the estrogen receptor, or ER, can help a bunch of cells move together again, can invade our bodies better or form colonies or have this independent growth outside of the typical growth of what happens in prostate cancer. All in one.
So the gene expression thing is like that, cancer basics. So when the gene expression goes wrong and these cells start like mutating.
Yeah, that's a good way of thinking about it. Or the gene is going to be amplified more. So imagine, there's a number of different things that can happen in your genome when it comes to a cell being like, I'm normal versus being cancerous. You can have amplification. So typically in prostate cancer, AR is going to be amplified. So instead of just the standard AR just getting activated at a set number of times throughout the day, it's like a parent going up to the child's like, don't touch that, and it keeps touching, so you're just like, all that. And so that's one thing. Other mutations can occur where different target genes are also being upregulated. We're not going to talk about it because this is in plain English. We don't have to get into it. But the gene expression can be affected to a point that causes the cells from going normally to being, I'm ready to invade. I'm ready to be independent. INDEPENDENT.
So when we're talking about androgen independent prostate cancer, we're talking about a kind of cancer that no longer needs that kind of like kid pushing a button a million times to be cancerous.
For sure. The more we talk about this, it imagines like Dexter's Laboratory and DD a little bit, but everyone, if you don't know what that is, I don't know what you've been doing when you were a kid. But yeah, so this particular cell line, PC3s, it is androgen independent. However, there's some debate around it, but it's usually a collective understanding that this particular cell line does not need androgens to grow. Matter of fact, I've worked with them a number of times and put them in different media that doesn't have any not growth factor, but any androgen, and it grows fast. They grow like weeds, as my advisor would say. So everyone who's listening, when it comes to estrogen receptors, just remember from this entire conversation, one of these things does not belong here, and yet it's working well in terms of affecting a cancer cell line's ability of being cancerous. And so that actually leads to our first figure. So the first figure we're actually going into is data that this particular group found in terms of using an assay called the wound healing assay. So I don't know, when I first started reading this paper, all I could imagine was the Monty Python point of having, 'tis a flesh wound. And it's essentially a flesh wound. They took a 24 well plate and seeded the PC3 cells in it. They added myosin C. The reason being is that they want to avoid cell proliferation, which for everyone at home, cells are going to grow regardless. You can hit them, you can hurt them. Cells are growing constantly. But if you truly want to understand how migration is happening, which is when cells are apart and they come back together again and see each other, be like, hey, how are you? You need to add some factors within your media in order to avoid cell proliferation. Otherwise, your data is going to lie to you and just be like, oh, it did work. It's like, well, that's because more cells just filled in where it left. So you take the 24 well plate, you take a pipette, you just take a tiny little prick down the line of the different wells of the plate. It's like a tiny flesh wound, and let it grow for 24 hours, depending on the different factors that you're adding into it. So in this data, you have a comparison of when you first gave the wound and 24 hours after you gave the wound. For the first part of the figure, we have we have a control. A control is essentially what is going to be what you're comparing in terms of when you give your experimental features of your entire experiment. So in the control, I give the wound, I don't give the wound, but our lovely scientists gave the wound to these cells. 24 hours later, there was a little bit more of a closed gap. Then we give E2. Does anyone know what E2 is?
This is the estrogen?
Winner. I don't know if you eat chicken, but chicken dinner. 0 hours, they gave the wound, and they were able, after giving, well, not even after giving the wound, they gave the wound in media that would have E2, which is just estrogen in this case. And after 24 hours, there's an even more closed gap, which speaks more to estrogen signaling pathways being activated, moving fairly quickly. and activating just overall signaling in order to close the gap that the wound created. And that was only at 0.1 nanomolar. They increased it a little bit more, and in a great way fashion, there was more of a dose dependent change in terms of giving the wound. So with more estrogen around, the cells were more excited to get things moving and pumping and migrating back together. So that was their first part of the figure. Pretty standard. And they even include a pretty graph on the right side. So next, they decided to look into other agonists or different activators that are going to activate the estrogen receptor. And particularly, they're looking more into the estrogen receptor beta. So for everyone at home, there are two different types of estrogen receptors. There's alpha and there's beta. Their difference in terms of how they interact with each other is estrogen alpha can interact with estrogen alpha, and beta can interact with beta, and alpha and beta can interact with one another. However, it's been found estrogen receptor beta can actually be suppressed in certain scenarios when it's around alpha at times. It all depends on where the cards fall. So
endocrinology is just absolutely wild. There's literally so much happening in your cells at all times. Trying to isolate one thing and just study that one thing is absolutely insane.
It gives me proof that I will always have a job. In terms of this particular part of the figure, we're in B now. This time they're looking at agonists and antagonists. So control, you give the wound, 24 hours later, not much of a close, which we expect just because cells aren't being affected as much. Give DPN or ERB, or just going to be estrogen receptor beta dash 041. These are known as estrogen receptor agonists. For everyone at home, an agonist, just think of that little brat that agonizes you. It's going to get on your nerves. It's going to make you move a lot and be like, oh, I want to fight you, or just move in this case.
I did want to say, as a person who deals with books a lot, that I was like, oh, an agonist. But we were talking about antagonists, and it's like, oh, I know what that is. But I'm more familiar with protagonists than I am with agonists.
So give DPN, you give estrogen receptor beta 041, and overall they end up closing after, well, not closing, but in 24 hours they give a tinier gap between with one another, which would we expect because they have similar activity to the estrogen 2 or E2 in this case. So then afterwards, they decide to let's add an antagonist in. So it's the opposite of an agonist. And in this case, they give PHTPP. I cannot pronounce the entire drug, so I will leave it at that. They add the antagonist to the agonist, and as would be expected, there will be a halting of what the agonist actually does within the wound healing assay, and overall keeps the gap a little bit open for both, at least for DPN. When it comes to ERB beta 041, there's still, some closure happening within the wound, but it's still a little bit wider than what you expect when you give estrogen receptor beta 041 alone. And then there's a graph for that too.
Sorry, I'm gonna, for the first figure, it was proving that estrogen is working to close this wound. Mm-hmm. And then for figure B, they're trying to prove that they're like estrogen like things are also working, but not as much.
No, they're doing like kind of a profile of these three things that can affect estrogen just to verify estrogen receptor is occurring. I actually looked into these DPN and ERB -041. There are a lot more going into the specifics of the estrogen receptor beta in this case. Because, if you're going to make the claim that the estrogen receptor beta is your major cofactor of closing this, you can't make that claim and then reviewers are going to be like, well, how are you going to specifically target estrogen receptor beta? So you need these two things. And actually, I think one of these, it's the ER beta 41 that have more an affinity for the estrogen beta versus estrogen receptor alpha in this case. So they gave us two scenarios to be like, in the first part, we're just going to give estrogen, something's going to happen. In the second part of this figure, we're going to give DPN and estrogen receptor beta 41 because we know it specifically targets estrogen receptor beta in this case. And so now they clarified, oh, beta's doing a wonder in PC3 cells in this case. And that kind of is even verified now with C, because with PPT, this is actually estrogen receptor beta and an estrogen receptor alpha agonist. And it still closes, but it's not closing in terms of like there's no major migratory changes like you would see with, say, estrogen receptor beta, which lets us know a little bit more. Estrogen receptor beta is the one to go through. So we were given flesh wounds in the first one. Now we're about to start a whole colony, no pun intended, with looking into colony formation assay in soft agar, which is essentially seeding cells in a gelatinous type of well and allowing the cells to grow in a 3D format. So typically for everyone, when you grow cells, there's different types. You have adherent and you have suspension cells. These are not suspension cells, these are adherent. So typically when you put the cells on the plate, they're like, oh, this plastic looks pretty nice. I'm gonna lounge here, and then they spread out. But that only speaks to how a cell's behavior is going to act in 2D, because you're looking at it, it's just spreading out. When they're in 3D, like it is in soft agar, they have a whole different behavior. And so now we get to see how they're going to form colonies on their own, which speaks more to their ability to just grow independent. And so they repeat the same conditions, just in a nasty gelatinous void.
I have a question. If these two ways of growing cells differ, what's the point of doing both? Like if you're telling me that like you want to check it out in 3D because they, you know, the way they develop colonies is much different than if it was kind of in a petri dish, right? With this 2-dimensional growth. Why do the 2-dimensional growth in the first place? Why not just be like, well, we care about 3D because that's how things work, so let's just jump into that? Or is there something about doing this kind of 2D growth that gives you more information or something?
No, for sure. So a lot of times when people go into the 3D space, the behavior of cells can change more towards being realistic towards the cancer that's being a part. So I'll give an example. She's on my thesis committee meeting. She works with breast cancer cells and has shown the difference in behavior in terms of what genes are going to be turned on and turned off when cells are adhering in 2D and when they're in the gelatinous space of 3D. So that's a benefit. One, because if you can find, you know, the caveats in different cells, you probably can beat people to the punch in understanding cells' behavior. The other thing is not a lot of people do colony formation in a just 2D format because it's possible. It's just not as good as if you were just doing it in a 3D space. So like I think when I first started in grad school, my PI was like, you should do this. I'm like, all right, let's do it in 2D. And it looked nothing like I thought it was gonna look. It's like, yeah, this just looks like a visual of what's happening in your wells when you're killing them. It's like, yeah, it's not really telling us the formation parts of things. Because you want to give, when you put them in 3D, you're giving the cells a chance and a lot of like different conditions to be like, now grow into this bump on this gelatinous thing. And so most of the time it doesn't happen that way, but in this case it does. And so that's some of the benefit.
Yeah, so like when we're talking about the wells in the first figure, those aren't colony formations. That's just like you put some cells down on there and you do a thing to them, and they're not really growing beyond that. They've got 24 hours to close the wound and then that's it.
Exactly, because, and this is going to be important just for everyone's general knowledge of just like, we're talking about cells we're talking about migration, we're talking about invasion, we're talking about anchorage independent growth. A lot of the time, the particular pathways that are involved, they don't really get a chance to shine. Because most people know cells, yes, they spread out, but they're also talking to friends and loved ones that are around them. Be like, hey, you should probably stretch that way, because I was about to call them a name, but we're going to leave it at that. But like communications being spread around in different cells. And so like you put a group of cells down on a plate, they'll start replicating, but as they replicate, they're communicating with one another. Let's move over here and stretch over to this clump of cells and see how that works. And that speaks more to its migration. Otherwise, you know, they can keep proliferating, but eventually they might run into each other like, I'm growing. No, you're growing. No, I know I'm growing. All that. You need that communication to regulate each other. Otherwise, your gene expression is going to change. Kind of similar to what we did in the beginning with figure 1. You have a control. There's a number of different colonies in this soft agar plate. You give estrogen at 10 nanomolar. They just went straight to it. Forget the 0.1. And the idea is you see visually, there's way more colonies. There's more colonies in that particular square versus the control square. And they can even graph it, at least on a bar graph. I even wrote here, E2 make thing go up like that. That's all I wrote. And then in B, they repeat what they were doing before with agonists. At least in this case, it's just going to be E2, but they give antagonists in this case. So they give the control, which has nothing. There is that set amount of colonies. And everyone, if I need to give a visual, just imagine a map or just a circle with a bunch of pins in it. And that's how you can imagine your plate. So in the control plate, there are just a little bit of pins all in your circle. You give E2, the amount of pins that are within the circle increase, which represents the amount of colonies that are being popped up in this case. Then you go into estrogen and PHTPP, because I'm not going to say that word. And remember, this is your antagonist that's being used for estrogen receptor beta. And once again, in this case, you give it to your colony, or you give it to the amount of cells that are forming colonies. And there's a drop, almost as close to, well, it's not close to control, but it's at least significant enough from a drop from estrogen receptor alone, or just E2 alone. Then they give MPP. MPP is actually an estrogen receptor alpha antagonist. In this case, they're doing a comparison between the two of how much of a drop do you get when you give an ER alpha antagonist versus an ER beta antagonist. And there's a drop there as well. It's significant enough in comparison to E2. And then they get both together. And there's even, it goes below, matter of fact, the amount of colonies that are there in comparison to control. Just to make their point that you can just inhibit everything in ER and you're good.
Is 10 nanomolar of estrogen like a quantity that would be seen physiologically?
I mean, I hate being that person that says it depends. You could see it. So the reason why I say that is because, and I'm only basing it off of my work with prostate cancer and not working with estrogen, but working with androgens. So like, that is it's within the concentration to be usable within experiments. I can say that for a fact, especially with reading different papers. A lot of people even use both, like in picomolar, depending on what they're going to be analyzing.
Well, I guess like because it's all on a spectrum, right? Like I guess Jamie's question was like how much estrogen is a person producing normally, and like if it was occurring in a person in a body, is the 10 nanomolar? No, molarity? Molar. Yes. I knew it.
That's a good question, actually. I'm not entirely sure. I'm also not entirely sure how you measure the amount, because the only way you probably measure in terms of concentration of your estrogen levels or your androgen level is if you use a different marker. So like I know for androgens, we use PSA. And in terms of estrogen, I'm pretty sure there's different markers that you can use to measure concentration to see if it's up to par. In terms of getting an actual like such and such is that this concentration, we need them up to here. Not gonna be as black and white as you would want. But I will say, in terms as far as how papers go and how the analysis goes, within right. Now, if they were like, bump that, we're going to millimolar, someone's dropping them. Someone's trying to cheat, essentially.
Someone's overloading the system.
Someone's overloading the system. And I don't know what their cause is. It might be for a good cause, but I will say, don't do that. That's something you do when you graduate and you're like, What are all the crazy experiments I've always wanted to do?
So for all the kids at home that didn't take college level chemistry, the nanomolar and the millimolar molarity has to do with concentration, and the nano and the nano and milli are I know the word I'm looking for, math.
Yes, math.
Degrees of.
They're a 6 log from each other. So for everyone, if you said millimolar, it would be 1 times 10 to the negative third. All right. And nanomolar is 1 times 10 to the negative ninth.
Okay.
So in terms of like biological amount of stuff you would have in your body, millimolar is actually huge.
I mean, it's big, but it also, you have to take into account. Do you work with cells, Jamie?
Yeah, I do a lot of cell neuro.
Okay. So wait, do you have to use a particular serum with your cells?
Yeah, we use a lot of like fetal bovine serum.
When you get that serum, all of the factors are within, say, nanomolar to micromolar. At least when you get it in that tube and it's processed or whatever. And imagine what happens when you add it into, say, your media. It drops tremendously. And so that's something we have to remember. Our cells are growing with not even a speck, like now I'm about to say all the factors of a speck. An eyelash hair on an eyelash hair on an eyelash hair. That's how small we need in terms of making these cells grow. And mind you, these are still PC3 cells, so they'll grow in anything. It's just how much is in there for it to grow.
Yeah, just like ambient nutrients and stuff.
Yeah, ambient.
That's because you don't want any ambient nutrients. Because you don't want to see whatever your serum is doing. You want to see whatever the thing you're adding is doing.
Mm-hmm. We went through E2 and giving antagonists. They also decided to do DPM, which again, ER beta particular agonist, and giving PHTPP, which is the antagonist in this case. And they repeat the same factors in terms of the fact when you give control, it's at a certain level of colonies, you give DPN, that goes up. You give the antagonist PHTPP, it drops. Once again, they do it with PPT, which from the previous figure, that is your ER alpha and ER beta agonist, it goes up. You give MPP, which is your ER alpha antagonist, and it drops again. So showing both factors can cause a drop in colony formation and overall affects PC3 growth.
So basically their experiment worked. They set out to show that the estrogen receptors are doing this thing, and they are doing that thing.
Yeah. In terms of the fact that within a particular pathway, which we'll get into soon, right? And for everyone, I think I probably misspoke. This is more so going towards invasion. We're talking about invasion type of stuff for figure 2.
Hey, in plain English listeners, Ralph here back again just to verify what happens in the cell invasion assay. So remember, we're working with PC3 cells, and we're actually going to seed these cells in a 24 well plate in this case. And in this plate, we're actually adding something that looks kind of like a communion cup, but in this actual cup, there are chambers. And in those chambers are these membranes that are coated with matrigel. And all you're looking within cell invasion analysis is when cells go through this membrane onto the other side, and that is determined as invasion. So after seeding these cells within these communion cups, and you add them into the 24-well plate, you'll then incubate them for an amount of time. I think it's about 48 hours. And you do it in the presence of each drug group. We talked about estrogen 2. We talked about DPN, PPT. And this is all done at 48 hours at 37 degrees Celsius. Afterwards, and this is with and without the drug that we're focusing on, which is PHTPP. After that incubation period, you will then take out the communion cup, which is just the chamber that you're using for the invasion, and you will then rinse with PBS. You'll fix the cells that you have, and then you eventually will have to wipe off the cells that are on top of the first part of the membrane. This is just counting as not cells that are not able to invade in this case. And once you wipe off those cells, on the bottom are the cells that actually invaded, and you get pictures. And those are the pictures you see in the figures that we've been talking about in this episode. Just wanted to clarify. Enjoy the rest of the show. Jamie's awesome. Maura's awesome. Alexa's awesome.
But in this case, we're talking about cell invasion. True colony formation is what we're getting into now.
So yeah, so there you said it. There was like three things. There was like invasion, migration, and what was the other one?
Anchorage, independence. Can a cell be independent on its own? We get into figure 3, and in terms of this, we're actually seeing colonies form from the one control. I don't like these pictures, you all, just because they're so bland. They're not bland, they're just like, here's a cell and watch it, boom. And for everyone at home, what I'm saying is more so, you get a better visual with certain assays. Like even the first assay we did with the figure 1. Wound healing is very visual. You just draw a line in the sand. In this case, draw a line in PC3s and watch it fix itself. Invasion assay, you see these cells within matrigel and you watch things pop up. In this case, I'm still seeing cells in a gelatinous thing with soft agar, but it's just floating and it's just gross. And it just it slowly spreads into becoming it looks like a city, does it not, Jamie, in this figure?
Yeah, you're making a little cell city.
Making little cell cities. So with A, once again, conditions are control, estrogen, DPN, PPT. With control, you get a visual of I'll just say 2 tiny little colonies that are just in like little tiny colonies that are just there for like a representative. You give estrogen, it goes up in size. You give DPN, once again, that's this particular agonist. That goes up in size as well. You get PPT, that's the estrogen receptor alpha and beta, and it goes up in size as well. And then with beta, they kind of repeat this again. So the first one with A, it was more so towards like the size of a colony that can occur. B is just how many colonies can you get out of this? And like I said, control doesn't really give us anything. It just gives us 1 in what they're looking at as a picture. Meanwhile, with estrogen, there's a total of like 3 different colonies that they're graphing. And then with DPN, still around 3. And PPT, they're still around 3 or 2.
Oh no.
They're that's okay though. I think their main point with this is that as long as it's bigger than what we get with control, it's happening.
Yeah, that's true.
In terms of these plots, like you're looking at like the amount of growth, right, versus kind of the number of them. That's my rough understanding of this. Is there like one that's more important than the other? Like, should I care more about the number or the size or neither? Or is it like the combination of the two that's more important? Like, why are they looking at both here?
It's definitely a combination of the two. And the reason being is, I mean, in the realm of cancer, especially with people that are investigating how metastasis comes about, the whole point of metastasis, and for everyone who doesn't know what mets or metastasis is, it's just when cancer spreads throughout the body from the original point of where it is. So if I have prostate cancer and it spreads to my bone, it metastasizes out there, which is why a lot of this is important, just because migration affects how a cell is going to move outside of its original location. Invasion definitely is going to make a move. And in this case, the colony formation. What you're mainly seeing with this is the capability of a cell to be like, I'm going to leave, and then I need to figure out how I'm going to survive on my own. And in this case, when it has the right conditions, E2, DPN, PPT, doesn't really matter. It can survive on its own in terms of growing in size, and it can survive on its own and having more other cells and other colonies form in order to build this city and environment to make the cancer be in a different place than where it was originally. You know what I'm saying? So like, one is never going to outshine the other, because I can give a lot of the colonies, you can have a lot of colonies out there and they can still do damage, and you can have one colony that's super big and it can still do damage too.
Super cool. Does this like relate to, and this might be like going too far, Does it relate to any degree in terms of how you'll like thinking about how people develop cancer and get diagnoses in the real world, and how it's sometimes hard to get an idea of where they are in the progression of the cancer? Is that kind of touching into this too? We're talking about like how when the cancer metastasizes, I probably said that wrong, but when that happens, it goes throughout the body, is like the complexity in terms of like they're both important and they interact and yada yada. Is that related to kind of like how people sometimes have a hard time, or it's sometimes harder to pick up on if a cancer is widespread or not?
Partly, and the reason being is like a lot of cancer, especially with prostate cancer, is a very slow growing cancer. So there's the benefit of being slow, so you have time to start diagnosing it correctly. But you also don't know when after you start giving treatment, when it starts getting resistant, it can switch up on you at any moment. And because of this, when it metastasizes, a lot of the time it could be too late because one, we can't detect where it's actually going, not where it's actually going, but more so just the Mets are still doing what they do in terms of metastasizing, but we don't know where they're going to end up in their last place. So if we go from the prostate, and typically it's going to metastasize to the bone, we don't catch the metastasis to the bone till after it's already happened. Which is why now a lot of doctors and clinicians, even my PI, they're trying to figure out ways to look into circulating tumor cells to see if there are any markers within the blood. Be like, this is metastasizing, or this is happening in the bloodstream and circulating. We need to add a certain drug so we can stop this from actually attaching. Because the whole idea is like you have a tumor, and then one of the tumors decides to be like, all right, I'm gonna go away and travel somewhere else. And it may take some people with it, it may not, but it's going and it'll go somewhere else. Same with breast cancer.
I guess my question is, so like we talk about, there are all these different kinds of cancer that affect different cells. So when it metastasizes, is that it changing to a different kind of cancer? Like if it goes into your bones, it's not bone cancer.
It can be, unfortunately.
Don't the
Oh,
cells like maintain some profile, like you're able to biopsy them and be like, this came from the primary tumor?
no, you can, but the instant it attaches to a different site, it's going to maintain that profile, but it's also going to pick up a new profile. It's like a filter, literally original, but I'm adding some stuff to me. So, like, that's the whole point with, especially when it comes to different prostate cancers that, in prostate cancer, it switched from being naive, so it can be affected by drug, and then it gets resistant. But there are certain factors that are upregulated that contribute to a cell's growth and invasion. All right, we're heading back to 4, and we're going to something we're familiar with: wound stuff. We're causing more wounds, everyone. So, but in this case, we have a special guest coming in. So, everyone, we have different categories. We have control, we have DPN, and we have a certain drug called PKF 118 to 310 with DPN. Does anyone know what that is?
Absolutely no clue.
You all definitely read the paper. Y'all can tell me what it is. I hope y'all read the paper.
I did. I did do my homework.
Okay.
I do not remember.
Alexa for 50 points.
Please hold.
Oh, come on, you all. So PKF 118-310 is actually an antagonist, remember that word, that disrupts a complex in cells called the beta-catenin, TCF, LEF complex. So we talked a little bit about beta-catenin before. Beta-catenin is mainly going to be involved in cell adhesion, migration, and communication. Just allowing cells, like I said before, if we put them in a plate and they're spreading out and they see a homie over there, they're going to talk to that homie over there. They need beta-catenin. But during this time, beta-catenin action is involved in a pathway where it will go into the nucleus when stimulated and interact with TCF and ELF. TCF is T cell factor, and LEF is lymphoid enhancer factor. Disclaimer, I am not an immunologist by any of the means. But looking into those particular factors, I found that a lot of the time they act as HMGA mobility proteins, which all you need to know they play a role in chromatin extension. So overall, they can play a role in transcription. That's all you need to know.
They make you be able to make more genes or like turn more genes into proteins.
And that's kind of a lot of cells anyway. Like in terms of different factors, they like even estrogen, and I only describe androgen receptor signaling. Estrogen receptor signaling kind of goes in a similar fashion where ER alpha and ER beta combine together and bind to DNA, and a set of genes are on. This is the same thing. Beta catenin comes into the nucleus, binds to these factors that are on DNA. Genes turn on. Let's do transcription. Now, the reason why I bring this up in terms of this particular drug is that it is disrupting this particular complex. And by disrupting this particular complex in the scope of having DPN in the cells, this was their first indicator of estrogen receptor can play a role within communication with beta catenin pathway. You see this because in terms of control, in figure 4 for everyone, do the wound. Wound barely closes. Do the wound again in DPN. Wound closes even closer. But you do the wound and you give PKF, and I'm just going to say PKF. I'm not going to do all the numbers. Give PKF in the presence of DPN, the wound barely is going to close, which ultimately is going to relate to cells not being able to migrate to one another to close the wound, which means, in this case, estrogen receptor signaling that's going to beta catenin is being disrupted. So that was their first sign of anything. I think this is like I won't say it's my favorite, well, it is my favorite figure. This is their penultimate figure that's like, look at what we did. We disrupted something where ER is going to interact with beta catenin.
They're basically showing a hint at the mechanism. Like, why does this work?
So that like, is it that ER receptors, like the ER receptors are that's the estrogen receptors, whatever, acronyms, are using the beta catenin pathway, and like that pathway is being disrupted?
They're not showing, they're hinting that there is a possible mechanism. They're not telling us the mechanism flat out. Didn't even look into that for that paper, but they're mainly saying there is some connection here between the estrogen receptor and beta catenin, because when we give the beta catenin antagonist in a realm where estrogen receptor signaling is ramped up, something is being affected.
And so this is helping prove their point because
it's helping prove their point, in terms of what we've been talking about in this paper, is giving an antagonist that can stop the invasion, the migration, and the colony formation. So far, we've only been giving activation of all those pathways and then the suppression of all those pathways with the drug. But just because we give that particular drug, that only focuses in on the estrogen receptor. So we got that covered. But we never knew what the estrogen receptor was doing when it's going through an inhibition or if it's being affected in terms of migration. So what some people might do, and a lot of people these days do, they just look at what's happening in the genome when it's time to like activate everything. But in this paper, they're like, no, we're not doing that. So they either might have had like an idea of, well, if we're talking about migration and we're talking about anything with cell communication, let's just look at different pathways that play a role. Beta catenin is a big one in them. So in that case, they're like, okay, so let's not inhibit the estrogen receptor right now. Let's activate it. But we're going to inhibit the beta catenin pathway. And if there's a response there that is more conducive to what we've seen with estrogen receptor being inhibited, that's our ticket.
So basically, like, they're disrupting this larger, more quote unquote used cell pathway to like see that they don't, the drug doesn't have to be as specific as the estrogen receptor in order to do something.
Exactly. And all to just make sure that they're making connections between the two. It's kind of a risky move because they kind of went fishing. They took a pull, they already had estrogen, like we caught some estrogen, it's in the back of the truck. Estrogen receptor in the back of the truck. But let's see what else we find in this pool of why this pathway analysis that we're doing. And they cast it out, hope for the best, and they pulled out the beta catenin. They're like, cool, we found it. Now, mind you, definitely more studies need to be done, but this still is important to know. If anything, as Alexa has already saying, science is cool, but it's also scary.
So based on this finding, like what would be, I guess, from your understanding or your knowledge, like the next step? Like, okay, we found this, that kind of like fishing expedition worked. Like now what? Like, what does this mean?
If I were them, I would want to figure out if, one, go on a general search to see if estrogen receptor can directly bind or have a different connection with this beta-catenin pathway. And this would require a lot of Western blotting. Western blotting, for everyone out there, it's like barcoding for like cells. You're looking for a specific code. It's just looking for like presence. And maybe I say that is because if they say their claim is that estrogen receptor, beta-catenin pathway, they're mixed in. And I'm pretty sure there are studies out there that have shown this mix in already in different fashion, but specifically this complex, how does this play a role? Another thing they probably want to look into, and this is like, I'm just giving ideas of projects for grad students in the future. If you're focusing on the T cell factor and you're focusing on the lymphoid enhancer factor, it will be interesting just to try to see if you can specifically perturb those and see if that plays a role. Because eventually, or you can even delineate what each factor in their genes are going to turn on in this complex that's being created. So like T cell factor might, when it binds to the DNA along with the complex, it may have a set amount of genes that's going to be like, I want this, 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 and this. And same with ELF. And so if anyone out there that wants to get a PhD.
If there are any grad students listening to this, go to your thesis committee.
Even if it's not your subject at all.
Neuroscience, bump that. Go to it.
We love like crossovers between fields. I mean, like I love neuroimmunology and all of the like interesting things that sit at like the intersection of these two things that like actually people didn't think. This is a tangent that I'm going to cut out, but I think it's fun. People did not think that the nervous system had like any interaction with anything immune related at all. They're like, it's immune privilege. There's no immune stuff going on in the nervous system. None. Because like you don't get all of your like white blood cells that are in the blood. They can't go into the brain. So people assumed that there's just no immune system happening in the brain. And then people figured out like, the brain has its own kind of specialized cells that deal with invaders. Yeah, there's a whole ton of stuff you can do in neuroimmunology. So it's cool that it's like an intersection of fields that even a decade ago people thought just didn't touch.
No, you should definitely include that in the episode because one, I will be the first person to say I never stunted the brain in terms, and when I say stunt, I mean like I never really paid attention to the brain until I started studying prostate cancer. And the fact is the hypothalamic pituitary adrenal gland like axis, the whole thing is powered by the brain. Your whole like entire like gonad section, brain. Like forget what you think is really happening. The brain plays a big role. And I'm honestly, I wouldn't say I would go back to school and study neuroscience, but I will say as I go to like looking for a postdoc or whatever, I would love neuroscience courses because if you ever, and anyone never thought in terms of what I said, I'm not out of the job. Neuroscientists will never ever be out of a job because their whole thing is different than the entire body. Forget what you study. Brains change every day. Sometimes they choose violence. Sometimes they choose my anxiety. They change every day. It's stressful, but fun, but stressful. The fifth figure. So we looked at wound healing. We're back into cell invasion assay. Remember everyone, circle with a bunch of red darts all around it. This is just going to be what you're going to see. Cells within matrigel just popping out of blue. You get control, which is what you see to the left in that square. You activate it with DPN. And once again, you get PKF. The idea is that with DPN, you increase estrogen receptor signaling, and with PKF, you're dampening it. And that's what essentially you see in the visual, both the actual cells you see in the well, as well as the actual graph. I'm sorry, Jamie. You're gonna get all these puns. It's the last well, it's not the last episode, but it's the episode over December. Like, let me win.
Yes.
Yeah.
Fun holiday episode. All the puns.
All the puns. So it drops. Same with giving PPT. This is the estrogen receptor alpha and beta agonist. You give PKF, and it drops in comparison to say DPN and cool. DPN is still your estrogen receptor beta agonist. PPT is going to be both. And when you give PKF, it drops in the amount of invasion that's happening. However, it's significant enough, but it's not as good as when you give a DPN, which speaks more highly to the estrogen receptor beta pathway, if anything.
So to clarify my own understanding of this, what this is saying to me is that because adding PKF, your beta catenin pathway antagonist, on top of DPN, which is your estrogen beta receptor agonist, you get complete, like, all of the invasion goes away. You're back down to zero. Whereas when you add this beta catenin antagonist on top of PPT, which is both estrogen alpha and beta, you only get like half of the colonies go away. So what that's saying to me is that, like, the beta pathway is more involved with the beta catenin pathway than the alpha pathway. And so when you activate the alpha pathway, it's going through some other mechanisms as well to form colonies that's not being inhibited by this PKF.
That's why you're going to MD PhD school. Show out, do the dang thing. Yes, that's what it would tell me 9 times out of 10. Now, of course, this is within the realm of this particular assay, right?
Mm-hmm.
I would need way more information to work with this. It would be nice if they were consistent enough to, and this is not bashing them, this is just in general for more information, to repeat every experiment with those different conditions. So I would want the wound healing assay again, but to do it with PPT, with this drug too, just to see if migration's happening. Because my prediction would be, if that was the case, it shouldn't, or it would a little bit, but because of that gap where estrogen receptor beta is being ablated and estrogen receptor alpha is like, I don't know, I'm still gonna do me. Everyone, this is probably, I mean, I don't know, like, I enjoy papers like this because they're straight to the point. Like, we're gonna do this twice. We got wound healing, cell invasion, cell number, and cell size for just this realm. And then we have it with the drug that we're using for today. Easy peasy. And that's what the point of this last figure. We're looking at cell colony size and cell number once again. Instead of doing Matrigel with the cell invasion, we're using 0.35% soft agar. Once again, cells growing within a semi 3D space, right? So with control, you see what I see, not a lot happening there in terms of colony size, like tiny little dots. You give DPN, the colony size gets ginormous. Then you give your PKF and it drops. This speaks more to what Jamie already said earlier, with that happening within the respective of estrogen receptor beta activation. And then you switch over to colony number. I see plenty of colonies in the control, but I think I only see 3 there, at least what's being circled. But they also start doing it in terms of like decimal, so we're going to leave it at that. You get DPN, it increases to 5. But again, that's going to be what you're comparing the DPN to PKF. In this case, you get PKF with DPN, it drops tremendously, which I don't know why they did this graph this way. I'm sorry. You have your control is 3. You go up, you get 5 with DPN, and then you drop to 2 with PKF. And mind you, that's okay. It's just.
But now, but they've changed it. Now they're doing it as a percentage of the control as opposed to comparing raw numbers. Why have they changed it? Why?
That's what I'm saying. Like, you had a good thing going previously, and then you switched it up. And mind you, you can do whatever you want in your paper. It's your paper. But, and Jamie knows this, and all of you probably know this because you've done, I mean, Maura, you too. Consistency is always appreciated. Keep it consistent. And I should keep saying that because I know I'm not consistent. So I'm talking about myself right now. Okay. So they get PKF alone, and you only get 2 colonies. I don't know why I want to say it like that when we're talking about percentage, but they're still on par in terms of control, just below control actually, which probably speaks more to even in just regular growth versus stimulation, PKF may have a possible effect. But they didn't analyze it this way. They had different conditions for this. So lastly, B, instead of using DPN, once again, they use PPT, which is your estrogen receptor alpha, and estrogen receptor beta agonists. In this case, it increases in size once again, and you give PKF, and this time there is a drop. This is the caveat I was telling you about with looking into this, Jamie, in terms of the fact that, we looked at cell invasion and it does some type of ablation, but it can change with a different assay. So that's why I still say I need some information still. But also, I don't know the specifics in terms of other things they're looking at in this lab. But overall, you give PKF and the colony size drops. And when you do colony number, the same thing happens. Where you have control, you have a set number. Here we go again with these decimals again. PPT goes up, you get PPT plus PKF, it drops off significantly. And with PKF alone, same thing. So overall, what did we learn today?
So much. I guess my big thing was that I didn't really understand the different agonists and what they did. So it was super helpful to walk through those, and like I feel like I understand. I'm starting to associate different acronyms with different things now, with the right things.
Yeah, I'm totally on the same page. The acronyms threw me off. It took like a while. I had to keep writing out like, okay, this means this, therefore kind of like reasoning through it. But yeah, that was a really clearer summary than the one I had scribbled across my printed out article here. So
I think I covered everything. If you didn't learn anything from this, I will just say, when it comes to endocrinology, all steroid receptors have some type of role. Estrogen receptor can play a role in prostate cancer, just like androgen receptor can play a role in breast cancer. The question is, where and how much?
Before we end and have people like plug their stuff, I think it would be cool for each of us to say like something that we're excited to see in this like area of research in the future.
I think it's super interesting to me from my like very, I guess, naive understanding of cancer and like at the cellular level as well, before this episode, being like, I had it very kind of divided, right? I guess how most people do before they really understand it is like, these are the cancers that men have, these are the cancers that women have, and these are the hormones and cells, and that kind of like you kind of associate one and one, and realizing that that's only half the story, and there's much more happening there, and that it's not just like men cells, women cells, and like men cancer, women cancer, and it's much more complex to that. So I'm really excited about seeing this kind of crossover or kind of like mishmashing of everything together, and that there's actually consideration to more than just like the men and women cells and like, oh, it's prostate cancer, well, it's the men stuff, and being open to that and looking into it and really understanding it I'm assuming gives you a better picture of what the cancer is and how it develops. And I think that's really exciting, is to see that it's not kind of all in these little boxes, as either maybe it was never that way, or maybe that's just how I conceptualize it. But I think it's really exciting to see this kind of unboxing of things and exploration.
Literally, when people start talking about men's cells and women's cells, I'm just like, men can get breast cancer, right? And they'll be like, what? And I'll be like, yeah. Now, women can't get prostate cancer because y'all don't have
prostate. Yeah.
Trans women can get prostate cancer.
Speaking of that, I would be more interested to see, and like kind of not in this kind of in the same vein, but kind of like flip it and see like, do androgen receptors have an impact in breast cancer cells? Like one that's like a cancer that is more traditionally
like associated with estrogens, and see if like the opposite is true.
Literally, y'all said that, and I forgot there's like nature papers that talk about it a little bit. It's interesting because this is only like a blip in this type of research, because I think if you learn anything, there's a spectrum with all of this, because in different scopes of cancer, even the exact thing we talked about with estrogen and androgen, it can be used as treatment. It just depends on what the cancer is going to do. I mean, there's things called biphasic treatment where you give the drug and you give the stimulant of the androgen or androgen receptor, and then you just let the cancer figure it out. Just treated, but I'm stimulated. So I eat a little of this and a little bit of that, and eventually the cancer just like, okay, we'll figure it out, and it just shrinks while still maintaining it's like, because the whole point of that particular treatment is to avoid resistance. I think a lot of the time we're not thinking that way. It's like, we just want the drug, we want to get it fixed, and everything's done. Well, it can be fixed, but your body's also like has been used to this thing for a minute, and if it gets like it goes into remission, like anything can happen after remission. So you want to get the best scenario where your cells are like normal, like they were before, versus we were treated, but now we don't want to be, so we're going to be resistant.
I think I stand by like that's terrifying, but also really cool.
Yeah.
That people actually like that actually thought of that, and you're actually tackling that in that from that perspective again, like scary that or really cool. I don't know, both terrifying and exciting.
I feel like with these sorts of things, you kind of have to like, because it's so like awful on a human impact side, you kind of have to be like, but from a scientific standpoint, it's interesting. You have to like balance that, because otherwise you're just like, I'm depressed.
If anything, it empowers me every day to go to the lab and find something new, especially when cancer is never going away, but we can bring a better fight towards it. There's always going to be something, a caveat of cancer. I usually tell people cancers are just like us. In terms of cancer has more human abilities than we give it props for. It's able to adapt to anything. It's able to grow and change and be like, I can definitely resist this. I can say no to this, or to other things that might actually be impacted a lot or not.
On that note, does anybody have anything to plug? Like any stuff that they're working on? So Ralph, I know that you have a podcast. Do you want to talk a little bit about that?
I don't know if everything we talked about before is going to be in the episode, but as I was talking about before, I just put out my last episode for season 1 for Scientifically Sound. I run a podcast, everyone. It's called Scientifically Sound. It focuses in on giving more of a creative narrative towards talking about music and science all in one. If you dig botany, if you dig agriculture, this episode's for you, because we're talking about Three Sisters, which is an American agricultural trend, and tying it to vocal arrangement, because it's essentially the same thing. So check that out. It's out now everywhere. In terms of finding me or just following the podcast, you can find it at Scientifically Sound on Instagram, all one word. TikTok, same thing. Yes, I make comedic scientific things. This is what I do most days anyway. You can email us for the Scientifically Sound at gmail.com. And if you want to find me like as a person, you can find me at Ralph the Scientist, and the I on the side is like 3 I's, because I'm the third. But you can find me there, and I don't know, find me at my LinkedIn. I need a job.
Okay, Maura, do you have anything?
No, because I don't want any of you to find out my fan fiction handle.
All right, Alexa.
Yeah, sure. I recently started or am in the process of starting a podcast as well with a colleague / friend of mine. We called it Brainstorm Exploring Minds and Behaviors. And the goal of the podcast is to bring together different experts from different fields and different universities to talk about a common topic that they all tend to tackle from different angles. And actually earlier today, I recorded an episode on memory, which we hope to have out in some amount of time in the future due to editing. But if you want to keep an eye out for Brainstorm Exploring Minds and Behaviors, we're on Facebook, Instagram, we have a web page and email. But if you Google us, something should come up. And yeah, more episodes to come. So on like different topics that are normally thought as psychology or neuroscience, but kind of explored from a multitude of angles. So stay tuned.
Very exciting. And all those things will be linked in the show notes if you want to check them out. Highly recommend it. And this has been our 5th episode of In Plain English and the last episode for 2021. We have been discussing the paper Estrogen Receptors Promote Migration, Invasion, and Colony Formation of the Androgen-Independent Prostate Cancer Cells, PC3, through beta-catenin pathway, with our expert Ralph White and our guests Alexa Ruel and Maura Lydon. You can download the paper for free on our website at inplainenglishpod.org. Make sure to subscribe to In Plain English on Google Podcasts, Spotify, or wherever you get your podcasts so you never miss an episode. We will be on break for the holidays and won't have a regular January episode, but keep an eye out for a special compilation of fun conversations that didn't make it into the main episodes. We'll be back with our regularly scheduled episodes starting in February. Happy holidays, everyone.